A very good morning. I bid to Dr. Robert. My name is Shahirah binti Sabana, and I am going to present on my final oral presentation entitled Research Execution in Shalom Valley Park in Annam. So basically, I will be presenting on how, to be specific, an undergraduate UMS student can conduct a research study in Shalom Valley Park in Annam. So moving on to the introduction. So basically, Shalom Valley Park is a recreation park that is located in Inanam, Sabah. It is known for its clear and clean water. It is also rich with aquatic insects, making it a famous choice for any researchers or undergraduate students who wants to conduct a research study that focus on measuring the water quality parameters. Moving on to the guideline on how to conduct a research study in Shalom Valley Park. So, in general, any students who wants to conduct a field study or any researchers in any sites in Sabah, they have to obtain an access license from Sabah Biodiversity Center. So, in order to obtain the access license, the students have to fill in the application form that is available online on the SABC website, and they also have to submit some required documents to SABC. And then if the application is approved, if the application is rejected, the students may appeal to the SABC. And if the application is approved, the students will have to complete a statutory declaration form before they actually obtain the access license. And once the access license is obtained and it is confirmed that you are allowed to do a research study in Shalom Valley Park, then the students can proceed to contact the management of Shalom Valley Park to inform all the details about the research study and came up with an agreement, mutual agreement from both sides, from both the management and the students. And then after that, the students can proceed to the preparation, and after the sampling is done, the students will have to finish a report on the collected biological resources and submit it within a month, within 30 days after the collection is done, to the National Competent Authority and Competent Authority. And for the samples that are collected after the research is done, the students can keep it to themselves or offer it to the competent authority or dispose it in a manner determined by the competent authority and finish a disposal report. So moving on. So one of the common objective of the research that have been done in Shalom Valley Park is to collect and identify aquatic insects. It is because aquatic insects is a good bioindicator to check on the water quality level. So as Shalom Valley Park is famous for its clear and clean water, so collecting aquatic insects from the Shalom Valley Park is will determine whether the water quality in the park is very clean or not. Moving on to the sampling process. So in order to collect an aquatic insects, the students have to prepare some equipment, which is a kick net to catch the insects, a tray to separate the insects from rocks, dirts, and other constituents, a GPS map to record the exact location of where the aquatic insects is found and collected, 70% ethanol solution, specimen container or specimen jar, and a forceps. And then the types of samples that will be obtained by the end of the sampling will be aquatic insects that differ in terms of their size, their shape, and of course their growth level. Moving on to after the sampling is done, the students may have to exit the site. So there are a few manners that have to be watched out. It is the duration to leave site. So the students or any researchers have the maximum of 2 days to leave the study site. So we can allocate one day to reorganize all the equipment used and to secure all the collected samples, and another day to transport all the equipment, all the collected samples out from the study site. So once all the equipments are packed properly and all of them are transported back to the laboratory or anywhere, the students will have to make sure that there are no traces of sampling is left on the study site, meaning that there will be no traces of equipment, no traces of collected samples anywhere in the study sites. Make sure it is clean and back to its natural state before the sampling is done. And the tools of equipment have to be in a great condition also. So after the sampling is done, it is ideal to collect all the equipment or tools in one spot only, as you can see in the figure, in order to determine whether there is a missing equipment or tools, and also to ease the transport process. It's also ideal to check for any damage on the equipment or tools and record if there's any damage found. And of course, the equipments have to be cleaned properly, have to be packed properly and cataloged. How about the condition of the samples after the sampling? So in this case, in Shalom Valley Park, the samples that can be obtained is the aquatic insects. So the aquatic insects can be handled in two different methods, which is to store them in a jar filled with 70% ethanol, or for dragonflies, which is not suitable to be submerged in alcohol. If it is caught alive, it can be placed in a specimen jar, and if it is caught dead, it can be placed in a folded paper in order to avoid damage on their wings. So after the sampling is done and then the sample is handled properly, we will have to curate the sample in order to maintain the state of the insects to avoid any decay process from happening, and also to enable proper research done. So there are two ways to curate the aquatic insects. Firstly, to submerge them in an ethanol and then place them in a specimen test tube or specimen jar filled with 70% ethanol. This is suitable for soft aquatic insects such as larvae and juvenile. And for aquatic insects such as dragonflies, they can be pinned to display their wings in order to inspect their wing venation and so many other physical characteristics. So insects labeling is important. So basically, the label insects, they must include the insect's order name, family name, the location where they are found, can also include GPS, and the collector's name. So it's very crucial to use a pencil to write the labeling for insects that are to be submerged in ethanol, because if we use pen, the ethanol will bleed the pen ink. Then all the labeling will be erased. Post-curation, so this is specific for UMS students who are taking conservation biology. After the sampling is done, the curated samples can be passed to ITBC for research and collection purposes. And that's all from me. Thank you.